Well, first of all, can I thank Pedro for that generous introduction, and could I also thank Alfredo Martinez and Pedro for the invitation to come and talk in this summer course. I have to say that I was disappointed when I was walking from the hotel to here to find it was raining. Now, being from Britain, we're used to it raining all the time, but this is the third major city I have been in Spain over the last 5 weeks, the others being Leon and Burgos, and in all three it has rained and rained and rained. So I don't know what that means, but the world is clearly changing. As I'm the first speaker in this course, I thought that it was perhaps sensible for me to, in a sense, provide a general background in relation to obesity before we're moving on to the specifics of adipose tissue. So what I'm going to cover in these two presentations is, first of all, and let me start by this, is this working? Yes, it is. Let me start by emphasizing that obesity is the major nutritional disorder in the developed world, and that's something that I've been saying ever since I started working in obesity research in 1975. So what I'm going to talk about in these lectures, and what I'm not going to do, is what Pedro said I was going to do, is to tell you the answer to everything. I'm probably going to tell you the answer to very little, but I am going to tell you about some of the issues, some of the questions, and maybe just a few answers. So what I want to start with is to deal with what we mean by obesity, definitions. I then want to touch on incidence and trends. I want to talk briefly about the diseases linked with obesity, in other words, why obesity is important. And then I'll move on to adipose tissue, the organ of obesity. I'll talk a little bit about different types of fat cells, and then I'll talk in the second presentation on the secretory role of white adipose tissue and the secretory role of muscle, myokines, and crosstalks. So that's the framework of what I'm going to try and deal with over these two presentations. Now, I'd like to begin by telling you or reminding you that obesity is not a new problem. And just to illustrate that, this is a little stone statue which you can find in the Natural History Museum in Vienna. It's approximately 25,000 years old. So again, the message is that although we now have a major emphasis on obesity, it's not a new problem. This statue is actually so iconic that those who win one of the major awards of the International Association for the Study of Obesity receive a replica of that statue. Okay, so what do we mean by obesity? How do we define it? Well, we have a very simple World Health Organization approved, as it were, definition. It's based on body mass index, and I'm sure everyone is familiar with that, which is weight in kilograms divided by height in meters squared. And it's a very simple measure, of course, and that means that it can be used not just on a population level, it can be used in a doctor's surgery, and it can be used on a population basis too. And the definitions that are normally used are that if you have a BMI less than 18.5, then you're underweight. If it's, that should be 18.6 to 24.9, then you're normal weight. If it's 25 to 29.9, you're overweight. And if it's 30 or more, it's obesity. And if it's more than 30, depending on who you are, it's described as severe or morbid obesity or class 3. And if you use the nomenclature of class 3, then class 1 is a BMI of 30 to 34.9, and class 2 is 35 to 39.9. So those are, if you like, the internationally agreed definitions of obesity. There are two problems with them. One, of course, is that it's an approximation or approximate indication of the amount of body fat. If you are a weight lifter, a muscle builder, and you have a huge amount of muscle, then you may have a high BMI, which is not reflective of obesity, but reflective of that high muscle mass. Secondly, in some parts of the world, and particularly in Southeast Asia, the threshold for obesity is generally considered to be lower. But for practical purposes, and certainly here in the West, these are the generally accepted definitions of obesity. Now, how common is obesity? Well, I've selected some figures here for you from the website of the International Association for the Study of Obesity. China has a very low incidence of the order of 5%, but it's increasing rapidly. Spain, which I put here, and England have approximately the same level of obesity in the population. That is close to 25% of adult men and adult women. And that's pretty hard. Those, both Spain and England, are towards the upper end or at the upper end of the European figures. The winners or losers, depending upon your point of view, are those in the United States, where over 1/3 of adult men and women are now clinically obese on the definitions that I gave you in the previous slide. So obesity is a particularly huge problem in the United States. Now, I showed a slide of a historical figure 25,000 years ago in relation to obesity, but there's a general sense that obesity has been increasing rapidly in terms of incidence in most Western countries. And I'll just illustrate that for figures from the United Kingdom. This is the incidence of obesity in the early 1980s. Just 6% of adult men, 8% of adult women. By 2010, it was around 25% for both. In other words, there was an approximately threefold increase in the incidence of obesity in the United Kingdom over a period of less than 30 years. And that sort of change is mirrored in most advanced economies as well, including here in Spain. Now, of course, if obesity was merely a matter of aesthetics and image, self-image and body image and so on, then of course there wouldn't be such a major medical and biomedical focus on the topic. It's not obesity itself that matters, but it's the diseases with which it's associated. And what I've done on this slide is to list the major diseases which are associated with being obese. Type 2 diabetes, hypertension, coronary heart disease, certain cancers, and those cancers include colon cancer, prostate cancer, and breast cancer, osteoarthritis, and sleep apnea. So we have a range of diseases, the incidence of which are increased when individuals are clinically obese. But the one that generally receives most attention is type 2 diabetes. And if we look at some of the figures for diabetes, and this is from the UK, but in a sense it's representative again of most advanced countries. Being obese, having a BMI of 30, is linked to a greater than 10-fold increase in the risks of developing type 2 diabetes if you're a male. The risks are even greater if you're female, and the more obese you are, the greater that relative risk. So here we're seeing a quite dramatic linkage between diabetes and obesity. It's not a 10%, 50%, or even a doubling of the risk. It's 10 times or more once you have a BMI of 30. In the case of the United Kingdom, which has a population just over 62 million, we currently have 3 million people who are diagnosed as diabetic. And it's estimated that there are perhaps another 750,000 in the population who are diabetic, but this has not yet been diagnosed. So the general perception is that we have nearly 4 million diabetics in a population of 62 million people. In other words, more than 6% of the population. And that high incidence of diabetes, which is mostly type 2, is a consequence of the rapid rise in the incidence of obesity. And again, although these are figures for the United Kingdom, they're very similar in most other advanced economies. The incidence of diabetes, of course, is even higher in the United States, reflecting the even greater levels of obesity than we have here in Europe. Now, of course, politicians and health economists are concerned with this not just because of public health per se, but also because of the economic costs of the explosion in the incidence of diabetes. Again, or as a consequence of obesity. Again, using the UK as an illustration, our healthcare system, our national healthcare system, is currently spending in excess of 7 billion euros a year just treating the diabetes consequent to obesity. So it's a huge and rapidly growing drain on the healthcare system. And again, similar figures, I'm sure, are true here in Spain. So these sorts of issues are why there is now so much focus from a public health, both at a national and international perspective, on obesity and concerns to try and understand what it's about and how one might reverse it. Now, with that background and putting, I hope, obesity in context for you in relation to the scale of the problem and what we mean by it, let me move on to, in a sense, the organ that defines obesity, that is adipose tissue. Adipose tissue is, or its expansion is, of course, what obesity is all about. And as we'll see in the next few minutes, adipose tissue, white adipose tissue, is the main site of fuel storage in the body. And we now know that white adipocytes secrete multiple protein factors involved in metabolic regulation, and they're the source of endocrine signals to the hypothalamus, and they're involved in crosstalk with other organs. And that's what I want to touch on. But before that, I also want to talk a little bit about brown adipose tissue, which is the main site of adaptive heat production or thermogenesis in mammals. In the next few slides, I want to, in a sense, put adipose tissue in context in terms of how much we can have of it. This is a simple body composition slide here. This is a lean individual with a BMI close to ideal, 22, 23. Their total adipose tissue is of the order of 15 to 20% of their total body weight. But for an obese person, that figure rises once their BMI hits 30 to approximately 45% of total body weight. And as their obesity rises, of course, there's more and more adipose tissue. But what I'm trying to emphasize here is that when you're obese, adipose tissue is the major, largest organ in the body. And one of the implications of that is that the things that adipose tissue does is almost certain to impact on the metabolism and physiology of the body as a whole. How can it be otherwise if half or more of your total body composition is this adipose tissue? Now, another way of illustrating how much adipose tissue one can have is shown in this slide that I was given several years ago by Dr. Vicky Baracus from the University of Alberta in Canada, one of the previous institutions with which I have been affiliated. And it's basically a CT scan across the middle, as it were. And that CT scan shows, this is a male who's extremely obese, a BMI of 46. What you can see in this blue turquoise color here is the subcutaneous fat. What you can see here in the yellow is the intra-abdominal, the visceral fat, all the fat that is hanging around in the abdomen. And the green bits here are fat deposits within other organs, the other organs being the grey and the black in the slide. But what I want to emphasize to you in showing that slide is that if you look across a section of an obese person, how much adipose tissue there is, both subcutaneously and throughout the abdominal cavity. So again, the message is that when you're obese, there really is a lot of adipose tissue around. Now, this slide, of course, is not of people, it's of mice, and it's from a classic book on the anatomy of adipose tissue by Saverio Cinti from the University of Ancona in the middle of Italy. And what he's done is to dissect out the key or most of the adipose tissue depots, both from an lean mouse and from an obese mouse. And in putting this up, I want to illustrate or highlight three things. The first thing I want to highlight, of course, is that there are multiple fat depots within an animal, both subcutaneously and internally. Secondly is that when obesity develops, most, if not all, of the major fat depots get larger. So you have a almost universal expansion of adipose tissue mass. And the third thing I want to emphasize is that if you look closely here, whilst that's clearly the kidneys, you can see pieces or bits of fat that look very white, and you can see bits here that look very brown. So that takes me to the next point I want to emphasize. And that is that classically, we think of two major types of adipose tissue, white and brown. The classical role of white adipose tissue is fat storage and release. And of course, when people talk about body fat and adipose tissue, most of the time what they're talking about is white fat. The classical role of this brown adipose tissue, on the other hand, is the generation of heat. And that, again, classically, has been viewed in the context of thermoregulation, the regulation of body temperature, such as in animals exposed to cold environment, or in newborn animals that need to warm up when they come into a cold environment, having been inside the mother, or it's the system, classically, that is associated with the arousal from hibernation of those species of animal that hibernate during the winter period. So here we then have is the classical concept of two different types of adipose tissue with fundamentally different functional roles. If you're Saverio Cinti, you will see them as a single adipose organ, but that's now quite a controversial issue. If we look at what these tissues look like under the microscope, here's a classical view again of a brown adipocyte and a white adipocyte. So classical in the sense that the brown adipocyte has lots of fat droplets. These clear white circles are fat droplets. It's what's termed a multilocular arrangement of the lipid within the cell. The second thing that is immediately obvious with the brown adipocyte is that there is a lot of cytoplasm, and you may just be able to see these slightly dark-looking bodies, which are mitochondria. And I'll come to the mitochondria in more detail in a moment or two. Now contrast that with the classical picture of a white adipocyte. A white adipocyte doesn't have multiple fat droplets. It has a single unilocular arrangement of the lipid. It also has a very sparse cytoplasm relative to the brown adipocyte, And although it's not very clear from this particular slide, there are relatively few mitochondria. And again, as we'll come to in a moment or two, those mitochondria are not the splendid creatures that the brown adipose tissue mitochondria are. So that's the classical view of brown and white adipocytes. That whole story has become more complicated in the last 2 or 3 years, because a third type of adipocyte has been discovered or identified, and that is the bright adipocyte. Bright if you're in Europe, and particularly if you're in Sweden. Beige if you're from the United States, and particularly if you're from Boston. So we have this intermediate type of adipocyte, the bright adipocyte, having some of the properties and characteristics of a brown fat cell, but not being identical in every sense. And there are certain molecular signatures, and I don't want to go into a lot of detail on that, but there are certain molecular signatures now which one can use to distinguish between a white adipocyte, a brown adipocyte, or a bright beige adipocyte. The brown and bright both have a particular protein called uncoupling protein 1, which again I'll talk a little bit about in the next few slides. But a brown adipocyte is positive for this marker ZIC1. The bright adipocyte is positive for this marker HOX9. So we don't simply have to depend upon the visual appearance of different types of adipocyte to try and define them as white, brown, or bright. We have molecular signatures which allow us to do that in a way which is, of course, more objective than simple visual appearance. Another reason why visual appearance can be confounding is that if an animal has very little body fat, especially if it has been subject to a long fasting, then the white fat cells will shrink and the fat droplets within them may become multilocular, like a brown adipocyte, rather than continuing to be unilocular. Equally, a brown adipocyte in an obese animal or obese person may have rather larger fat droplets, indeed even a unilocular fat droplet, so it looks superficially as though it's a white fat cell. So molecular signatures are critical in terms of distinguishing between the different types of adipocyte. Now, I said a moment or two ago that I would return to the question of brown fat mitochondria. And here again is this is an electron micrograph of the mitochondria from a brown fat cell. Again, this is a classical brown adipocyte, and this is a classical type of picture of the mitochondria within those cells. Now, the characteristics which I want to underline or highlight to you are first that there are a lot of mitochondria. They're densely packed together. Second thing is that they are richly endowed with cristae. These little lines going across here, these are cristae, and they're packed with cristae. And thirdly, those cristae go right across the intramitochondrial space. So you have a very clear picture of the type of mitochondria that you see in brown adipocytes. They're also large. Now, what that sort of mitochondrial structure tells you is that this is a highly active mitochondrion in terms of the oxidation of substrate. You would not have a mitochondrion like that unless you were concerned or determined to have a very high rates of fuel oxidation. Now, I said that the primary function of brown adipose tissue and of brown adipocytes was to generate heat. And this is a simple schematic of how heat is generated within brown adipocytes. I should emphasize two things. One is that brown adipose tissue is very richly vascularized. It has a very rich vascular system. Having a rich vascular system, of course, is important in terms of the delivery of substrates, the removal of waste products, but particularly the dissipation of heat, because the production of heat is the primary function of a brown adipocyte. The second characteristic that I want to emphasize is that they're also densely innervated by sympathetic nerve endings. And it's the sympathetic nervous system that drives fundamentally the process of heat production in the brown adipocyte. So noradrenaline or norepinephrine, if you are the other side of the Atlantic, is released from sympathetic nerve endings. It binds to a beta receptor, and this is predominantly a beta 3 adrenoreceptor on the plasma membrane of the brown adipocyte. There is an activation of the hormone-sensitive lipase via cyclic AMP and a series of protein kinase, and you have the classical breakdown of lipid to fatty acids. Those fatty acids are oxidized in brown fat mitochondria in much the same way that they would be oxidized in mitochondria of other fat-consuming tissues. But what is different about the brown adipocyte, what is different about the mitochondria within the brown adipocyte, is that instead of harnessing the proton gradient that is generated during substrate oxidation in mitochondria to the synthesis or production of ATP, brown fat mitochondria short-circuit the mitochondria so that instead of producing ATP, they generate heat instead. And the mechanism by which they can do this is essentially because of this protein, uncoupling protein 1, which is located in the inner mitochondrial membrane. And you can think of this process essentially as acting like an electrical short circuit. So instead of getting ATP out of the oxidation of lipids or indeed any other substrate, all you get is heat because you punched a hole in the membrane and the proton gradient is dissipated instead. So this uncoupling protein is a key diagnostic also of a brown adipocyte, or, as I said in a previous slide, of a bright adipocyte too. Now, uncoupling protein 1 was discovered in the late 1970s by Daniel Riquier from Paris, but 20 years later it was found that there are a family of these uncoupling proteins, although only this particular one seems to be directly involved in thermogenesis in the classical brown adipocyte manner. Now, just to illustrate to you how important this uncoupling protein is, what I've shown on this slide, and it's an old slide as you can see, is how if you put a small mammal in different environmental temperatures and you adapt it or acclimate it to cold, and you look at the changes that are taking place in brown adipose tissue, well, the tissue looks browner, there's more mitochondria, there's increased vascular supply to the tissue, but there is also a large increase in the amount of this uncoupling protein. And this a large increase in the amount of uncoupling protein is about the process of trying to generate more heat in order that the animal can both adapt and survive, or survive and adapt to low environmental temperatures. So this shows what happens if you keep rats at either 29 degrees, which is what is called the thermoneutral temperature, the temperature at which they don't have to generate any heat for the maintenance of body temperature, or if you keep them at what we normally regard as room temperature, around 21, or at 13 or 4 degrees, you can see that there is a stepwise increase in the total amount of uncoupling protein in the tissue, and indeed for a rat at 4 degrees, there's more than 100 times as much of that protein as there is in a rat at 29 degrees. In other words, its capacity to generate heat by thermogenesis in brown fat has been very substantially augmented. Now, from the late 1970s, there was an enormous interest in the obesity field in the idea that brown adipose tissue was not only involved in the maintenance of body temperature, but it was also involved in the regulation of body weight and obesity. And the central idea was that obesity is a result of either of inactive brown adipose tissue or insufficient brown adipose tissue. And this concept was perhaps one of the dominant ideas in obesity research for roughly 15 years, from the late 1970s to around the mid-1990s. And a lot of animal studies on rats and mice, were done in which the activity of brown adipose tissue in terms of its ability to generate heat were examined in different physiological, nutritional, environmental conditions. And what this slide does is to summarize many of these key results. So brown adipose tissue thermogenesis is increased in animals in the cold, as I showed in the previous slide. It's increased if animals are overfed, and these were some very important findings, particularly by Mike Stock and Nancy Rothwell in the UK, that showed that if rats voluntarily overate through consuming what was termed a cafeteria diet, that is a diet made up of human foodstuffs rather than the little pellets that rats and mice are normally fed. In other words, you give them cheese, you give them biscuits, you give them pate or whatever, they will overeat. But instead of becoming obese in the way that you would expect by virtue of that overeating, what happened was that they dissipated at least some, in many cases almost, or in some cases, almost all of that extra food energy that they took in, and this was associated with an activation of their brown adipose tissue. Other situations in which there were increases in brown fat consistent with changes in energy flux were consuming a polyunsaturated fatty acid diet, on low protein diets, with cancer cachexia, the weight loss and increased energy expenditure associated with certain types of cancer, through the administration of catecholamines, and also following arousal from hibernation. On the other hand, in obesity, and a number of different animal models of obesity were examined, such as the genetically obese obob mouse, the genetically obese Zucker fat mice, mice made obese through feeding high-fat diets, and so on and so forth. Fasting, hibernation itself, lactation, diabetes were all associated with reductions in the activity of brown fat, consistent with the concept that brown fat was playing a role in whole body energetics, and not just simply its regulation, that is, and not simply as a function of the response to cold. Now, although that idea was deemed to be a very powerful idea in small animals, there was enormous skepticism in relation to whether or not it was relevant to humans. A lot of people, a lot of magazines, articles jumped on the idea that human obesity was a function of a reduced activity of brown adipose tissue. But actually there was very little evidence in support of that idea, and the idea essentially died away from the mid-1990s until 3 or 4 years ago, as I'm going to show you. And the issue in relation to brown fat and human energetics, or human energy metabolism, was, first of all, fundamentally, is brown adipose tissue actually present in humans, or is it something that's really for small rodents exposed to the cold, newborn animals also exposed to the cold, hibernating species? Second, if it's present, is it thermogenically active? And can it be recruited? That's to say, can you increase the amount of it and its activity? Further questions were, is thermogenesis normally a significant component of energy expenditure in humans? And finally, following stimulation and recruitment, can brown fat provide a therapeutically important increase in energy expenditure? Now, by the mid-1990s, the answer to most of those questions was no. Brown fat was deemed not to be an important issue in humans, other than in newborn human babies, where if they are exposed to the cold, and of course most of them are not, certainly in the West, then it was really an irrelevant issue. But that all changed in April 2009, when there were 3 papers published simultaneously in the New England Journal of Medicine. And what these 3 papers did was to provide very strong, convincing evidence for active brown adipose tissue in adult humans, and also that it was adaptive, or its activity could change according to circumstances. And what was key about these series of studies is that they used a technique that had been, of course, a technique of nuclear medicine, which is PET scanning, positron emission tomography, and they coupled with that with fluoxydeoxyglucose markers to be able to map rapid glucose uptake in different organs of the body. And when this was combined with looking for uncoupling protein, definitive evidence for active brown adipose tissue in adult humans was shown. What this slide shows is one of these 3 studies, and the dark areas is where the fluorodeoxyglucose is being taken up. And you can see in subjects exposed to mild cold, there are a number of areas which are showing very intense blackening following or during the PET scanning. But in subjects maintained under thermoneutral conditions, 28 degrees, there is very little or very few of these black areas. So this technique was strongly suggestive that there are active sites of brown adipose tissue in humans. But what really finalized the issue was that biopsies, or in effect biopsies, where those tissue areas with high uptake of the labeled deoxyglucose were examined immunologically for the presence of uncoupling protein, and the 2 sites coincided. So we had, from 2009, very clear evidence that active brown adipose tissue was indeed present in many adults. And a number of studies using this technique of nuclear medicine have been done since that time, and 2 of them are illustrated here. This shows the amount of brown fat activity in relation to the amount of body fat. It's from the group in Maastricht in the Netherlands, from Van Marken-Lichtenbelt, and you can see that as the amount of body fat rises, so the brown fat activity falls. And this is a study from the Severio-Cinti group, which shows that the number of brown adipocytes falls with BMI. So now we're in a situation where there is good, strong evidence that brown adipose tissue is indeed present in adult humans. It can be stimulated by factors such as cold, and the activity is reduced with increasing BMI and age. And what this has done is to reignite the issue of whether or not brown fat is potentially a route for treating obesity. I'll say from the outset that I'm personally skeptical, but there are certainly a group of people who are pushing very hard and very firmly this idea that brown fat is again a key target for anti-obesity therapy. And indeed, there are European Union programs that are being funded on the basis of this concept. I'm the chairman of the Scientific Advisory Board, one of them, so I know this very well. So what are the options if you take this idea that you may be able to treat obesity through targeting brown fat? In other words, the renaissance of this idea of 20, 30 years ago. Well, the approach that has been used up until now was the idea that you went for direct activation of brown fat cells through the beta-3 adrenoreceptor, on the cell membrane by trying to design selective beta-3 agonists. For a variety of reasons, this never proved very successful. Alternative routes are that in some way you have a selective stimulation of the sympathetic innervation to brown fat. More recent ideas is that you can potentiate or you can increase brown fat development by a browning of white fat. And I'll talk just briefly on the browning of white fat in the second presentation a little later. And a final possibility that some have raised is that you can have stem cell therapy. So again, I want to emphasize two things. If you look at the literature, you look at debates in the obesity field, the concept that targeting brown fat is very much back on the agenda, but there are a number of us who remain skeptical as to whether or not that is an appropriate or realistic approach. Certainly, treating obesity becomes incredibly high-tech if we're talking about stem cell therapy in order to increase the number of brown fat cells. So, that's brown adipose tissue. I think I'll talk for maybe just a short bit more before stopping. So there's the opportunity either for any immediate questions that you might have, but also to give you a pause, because you've got 4 one-hour presentations, and if the 2 speakers each speak 4 hours without giving you a few minutes to reflect and breathe in between, you will feel completely saturated and overwhelmed. So I think I will just say 1 or 2 more things before closing or pausing for those reasons. I've made some emphasis or play on brown adipose tissue and thermogenesis, and I've talked essentially about pretty high-tech solutions that some people envisage for the treatment of obesity. But I think we also have to come back to earth, so to speak. And back to earth means that fundamentally obesity is a problem of the regulation of energy balance. Whilst genetics have something to tell us, quite a lot to tell us, the big increase in the amount of obesity or the incidence of obesity that we've seen over the last 2 or 3 decades in most Western countries is, of course, not a reflection of changes in our genetics. It's about changes in lifestyle, with the development of what many call an obesogenic environment. And that, in a sense, brings us back to this fundamental energy balance equation. You can only get obese, whatever is true about genes or anything else, if your energy intake is in excess of your energy output. There has been enormous interest in trying to unravel the neurofactors, the peripheral signals, and indeed the neural circuits involved in the control of appetite. And there's been a lot of interest in trying to unravel the components of energy expenditure, with again brown adipose tissue being one of the key sites of adaptive thermogenesis. But the organ or the tissue which buffers the whole process is white adipose tissue. By definition, if your intake is in excess of your expenditure, then your white adipose tissue mass expands. If your intake is below your expenditure, your white adipose tissue mass falls. And I'm going to stop at that point because my second presentation will focus specifically on white adipose tissue, the properties of white fat cells and how they differ from brown fat cells. I'm happy to try and answer any immediate questions you've got, or you may want to leave them to the end of the evening session, or you may simply want a 10-minute breather before the second speaker comes.